Hello, my name is Daniela Balslev. I am a lecturer in the School of Psychology and Neuroscience at the University of St Andrews in Scotland. I have been asked by the organizers of the International Society of Proprioceptive Disorders to speak with you today about ocular proprioception and attention. It is a great pleasure to talk about this research. My lecture today will focus on the anatomy and function of the proprioceptive system in the extraocular muscles in healthy humans. I hope you will enjoy the lecture and that some of you will think about disease mechanisms in a new, different way based on the material that I present today. So the talk is in three parts. In the first part, I will focus about the anatomy of the ocular proprioceptive system, from the receptors in the extraocular muscles to their cortical projection in the brain. I would also mention some exciting new results from my lab that show an ocular proprioceptive projection in the oculomotor nuclei in the brainstem and why this is important. In the second part of my talk, I will speak about a relatively known function of the ocular proprioceptive input, and that is to contribute to locating visual objects relative to the body. And I would also take the opportunity to speak in more details about the methods that we have to study this modality. I would like you to appreciate already from the beginning that it's rather difficult to stimulate the proprioceptors in the extraocular muscles without triggering an oculomotor command. And finally, in the third part of the talk, I would introduce the function of ocular proprioception in visuospatial attention, which is a rather recently discovered function. I would also deep into pathophysiology to look at a connection, a possible connection between a disorder of gaze and an attention dysfunction in patients with spatial neglect. So let's begin. The eye globe is moved in the orbit by extraocular muscles, and in these extraocular muscles, we know for sure there are receptors that respond to stretch. These receptors are proprioceptive receptors. Now, the nature of this receptor is rather controversial. So one proposal is that the muscle spindles, which you probably are familiar with from the skeletal muscles, also act as proprioceptive receptors in the extraocular muscles. Muscle spindles are present in human extraocular muscles, but rare in lab animals like cats and macaques. And one more thing, the muscle spindles seem to include normal contractile muscle fibers. For this reason, it is unclear if they can respond independently from the rest of the muscle to act as a stretch receptor. Now, all this means that animal models and human skeletal muscles are less suitable to understand the function of ocular muscle spindles in humans. What other possible receptors are there in the extraocular muscles? Some authors have proposed that palisade endings, which are specific for the extraocular muscles and do not occur in any other muscle in the body, might function as somatosensory ocular proprioceptive receptors. The most attractive feature of these receptors is that they are present in most vertebrates. Now, the problem, however, is that they exhibit characteristics of both motor end plates and sensory receptors. So For this reason, it is not clear whether they are oculoproprioceptive receptors or the terminal of the motor neuron. My interest in oculoproprioception was triggered by this paper from Michael Goldberg's lab, published in 2007. This is because this is a very elegant demonstration of the projection of the oculoproprioceptive input in the somatosensory cortex of the macaque. Let me go through their experiment. So, they dip an electrode, or rather several electrodes, in the depth of the central sulcus to record from an area located in the depth of the central sulcus, area 3A, that is known to receive proprioceptive input from the skeletal muscles. And so this was the best bet of recording neural activity related to ocular proprioception. During the experiment, they had macaques fixing their gaze at various locations in the visual field. So they identified neurons that were sensitive, or that liked a particular direction of gaze. For instance, the neuron pictured in panel B responds maximally by increasing its activity to targets that are located at 120 degrees. And all directions in the visual space were represented in this area that the researchers placed their electrodes. And something interesting that I will come back to when talking about transcranial magnetic stimulation later on, the firing rate, so the intensity of this neural activity, scaled with the eccentricity of the target. So the further from the center a target was, the higher the firing of the neurons. So the neural activity that coded the direction of gaze and the amplitude of the gaze was very likely to be proprioceptive, but it is not certain, because for the monkey used active eye movement, so one could in principle see the effect of the motor command. For this reason, the researchers did a second manipulation, and that was to look at the activity of these neurons while administering orbital anesthesia. So they essentially abolished the sensory input from the extraocular muscles and looked what happened with the activity of these neurons. And as expected for a signal that originates in a periphery, these signals were abolished by the block. So you see here high firing rate in the neurons before, high firing rate after, and a silencing of the neurons during the orbital block. This demonstrates that the macaque somatosensory cortex receives a projection from the extraocular muscles, and then this projection is proprioceptive. How about humans? Could we find evidence for an oculoproprioceptive projection in the human brain? And for that, colleagues and I conducted an fMRI study. The task we used for functional magnetic resonance imaging is inspired by the task designed by Ilg and colleagues. So for this task, one pushes very briefly and gently on the eyelid like this. This intervention, when done in complete darkness or on a closed eyelid, causes a movement of the pushed eye in the direction of the push, whereas the other eye is still. We know that because Ilg and colleagues have recorded eye traces with a very sensitive eye tracker when participants did this task in complete darkness, so with the eyes open in complete darkness. So we thought that we could take this task in the magnetic resonance imaging scanner and see the brain areas that are activated by passive eye movement. Now, because this is a rather complex task, so it involves not only a stimulation of the extraocular muscles, but also a hand movement to push the eye, so the participants push their eye themselves, and also some tactile stimulation. We had control conditions to get rid of these additional sources of activation to isolate the proprioceptive input. So for instance, we wanted to see brain areas that were active during both active eye movement and passive eye movement, but not during tactile stimulation of the right eyelid. And we identified this projection in the somatosensory cortex as well as motor cortex of both hemispheres. Now this is good news because it replicates the results in the monkey and shows that in the human brain we have a proprioceptive projection from the extraocular muscles as well. Another area that is controversial is the subcortical pathways that the proprioceptive input from the extraocular muscles travels. Fiber tracking studies from the extraocular muscles have identified an ocular proprioceptive projection in the trigeminal nuclei via the ophthalmic branch of the trigeminal nerve. An alternative proposal is that the proprioceptive input may also reach the brainstem oculomotor nuclei. This is an attractive proposal because it would show that the extraocular muscles are similar with the skeletal muscles. As you probably know, in the skeletal muscles, a stretch of the muscle stimulates the proprioceptive receptors and triggers a contraction via a monosynaptic reflex. This is the classic stretch reflex in the skeletal muscles and is, for instance, responsible when we carry a heavy load, like a heavy shopping bag, that we don't need to concentrate and all the time maintain our muscle contraction. This is something that the spinal cord does for us via the stretch reflex. Now, the problem with this hypothesis is that in the skeletal, in the extraocular muscles, the stretch reflex has not been observed, at least in some species like macaques and cats. So it would be interesting to see whether in humans we can find any evidence of a stretch reflex to support the hypothesis proposed by Lienbacher and Horn. To investigate this hypothesis, we used the same task that I presented before, but this time looking at the brainstem oculomotor nuclei. Now, imaging the brainstem is much more difficult than imaging the cerebral cortex. And this is because in the brainstem, the nuclei are very small, just a few millimeters. And also because these structures are very close to pulsating blood vessels. Imaging the brainstem requires special methods, and therefore among our co-authors for this study were two physicists, Lucas Priba and Jennifer McFarlane, who designed the data acquisition procedure. So the data analysis was the same as for the previous study, and we focused on the brainstem oculomotor nuclei. Before I show you results, I would like you to think about what you might expect to see if a stretch reflex exists in the extraocular muscles. So, we stretched the right lateral rectus muscle. So we had participants pushing on their right eye globe towards the nose. So I'm saying pushing on the right eye globe, but they actually placed their index finger on the eyelid. So this is not invasive at all. So lateral rectus muscle, the lateral rectus muscle stretched. If there was a stretch reflex, we should see activity in the brainstem nucleus that innervates the lateral rectus muscle of the right eye. And I don't know how fresh your eye anatomy or eye neuroanatomy is. I had to look this up. The muscle that the the motor nucleus that innervates the lateral rectus muscle for the right eye is the right abducens nucleus. So if there is a stretch reflex, we should see here activity in the right abducens nucleus. And we do not see activity in the right abducens nucleus. We saw activity in the left abducens nucleus, which innervates the lateral rectus muscle for the other eye, and the same left oculomotor nucleus, which again innervates the left eye. So what we found is that indeed the oculomotor nuclei are activated, but the pattern of activity is not characteristic for a stretch reflex. Now, because our results, imaging results, were not entirely as expected, we were curious to see what is the origin, so which brain structure drives the activity of the motor nuclei that innervate the left extraocular muscles, and also to check that indeed behaviorally we don't see any movement of the left eye. So this is what I'm going to show you in the next slides. To look for brain connectivity, functional connectivity, so effective causal connectivity, that is brain areas that drive the activity of another brain area, we used a method called psychophysiological interaction, in which we identified brain areas whose activity correlates with the activity in the left oculomotor nucleus during the passive condition, but not in the active condition, to find out where does this activation of the oculomotor nucleus during the passive condition comes from. And we found an increase of activity in the right trigeminal nucleus, which is where the sensory input, so oculoproprioceptive input from the right extraocular muscles are relayed. One caveat is that this is an indirect way of establishing causation. So ideally, one would want to somehow manipulate the neural activity in the trigeminal nucleus and see whether that has an effect on the oculomotor nucleus, but that would not be possible without actually conducting intracranial recordings. Okay, so let's now look at the behavior and find out whether the left eye was indeed still, as suggested by the paper by Ilg. So you see here example eye traces from one push trial. So the right eye starts with a baseline. The push to the eye is triggered by a sound presented at time 0, and you can see some milliseconds after the sound, the participant pushes the eye and there is about 9 degrees of movement to the left towards the nose, and then the elasticity of the tissue allows the eye to return to its initial position, and we see a second resting baseline. I have color-coded the the time interval where the eye is pushed, the baseline 1 and baseline 2, so pre-push and post-push baseline, to show you what happens with the left eye during those time intervals. So as you can see that, so first of all, please notice that the scale on this graph is much smaller than the scale here. So the left eye does not follow the movement of the right eye. It oscillates, but compared with the right eye, remains relatively still. And let's look at some statistics. So there's a clear difference in mean eye position for the right eye during the push and the two baselines before and after the push. We don't see a significant change in mean eye position for the left eye, and this is an average over 17 participants. In the standard deviation, however, we see that the left eye is less stable while the right eye is pushing. So that could explain the increased activity in the oculomotor abducens nuclei that move the left eye in opposite directions. However, something that is important is that the movement of the left eye between the two baselines is correlated with that of the right eye. So each participant completed 12 push trials, and you see the push trials from one example participant. The movement of the left eye from before the push to after the push baseline is on the y-axis, and on the x-axis is the movement of the left eye between those two baselines. And you see a good correlation between the movement of the left eye and the movement of the right eye. So looking across 17 participants, this correlation was significantly different from zero. So one could speculate that the function of the activity in the left oculomotor, left abducens nucleus that control the movements of the left eye is to keep the two eyes in alignment. So, in conclusion, the lack of activation in the right abducens nucleus in response to right lateral rectus muscle stretch suggests the absence of a stretch reflex. So, it's always difficult to conclude based on negative results, but this study adds evidence against a stretch reflex in the extraocular muscles. Ocular proprioceptive input from the right eye reaches the left oculomotor abducens nuclei via the trigeminal nucleus. This brainstem feedback loop is likely to contribute to interocular alignment. Based on these findings and taking into consideration hypotheses that have long proposed that ocular proprioception plays a role in ocular misalignment as observed in congenital strabismus, one could ask whether a dysfunction of the brainstem ocular proprioceptive feedback loop may play a role in pathophysiology. I would like to conclude from this entire section, the proprioceptive input from the extraocular muscles activates the oculomotor nuclei and the somatosensory cortex in humans, and the oculoproprioceptive receptors and the subcortical oculoproprioceptive feedback loops are atypical compared with the skeletal muscles. So I hope that you too can see that this is a very interesting topic and a lot of research remains to be done.